Good afternoon, everyone. I'm Felix Dobbs, co-founder and CEO at String Biosciences. And today I'm going to give you an overview on Broken String and our platform based around our technology INDUCE-seq for mapping genomic breaks. So DNA breaks are actually a super important feature of the genome that actually underpin a range of important diseases. These are things like genetic disease, the development of cancer and its treatment, and even neurodegenerative disease. And in the world of cell and gene therapy, we're now harnessing DNA breaks to edit genomes to confer therapeutic effect. And our platform actually addresses a major unmet need in the world of gene editing, really around the detection of off-target edits induced by gene editing. So, as many of you here will know, that gene editing is now poised to really be transformative in this space to bring essentially cures to previously uncurable diseases. But there's still this significant challenge that remains around the potential for off-target editing activity. And this is essentially where gene editing systems go into the genome and make alterations and changes in places that are unintended. The problem with this is that these kinds of off-targets can actually have pretty disastrous consequences for patients with significant adverse events. And so because of this, it's absolutely crucial that we understand where these sites are in the genome and understand the potential risks of inducing them. What makes this challenge harder from really a technical standpoint is that trying to find these off-targets is like trying to just identify a needle in a haystack when looking throughout the genome. Really, this needs a very sensitive next-generation sequencing-based approach to even get at this problem, but standard NGS tools are simply not suitable for doing this. Things like whole genome sequencing lacks the sensitivity to do this, and there are a range of other techniques out there that people are trying to use for this, things like single cell, and there are a whole raft of different off-target methodologies. But taken together, they don't have all the key features required really to address this off-target challenge. And that's where Broken Strings INDUCE-seq comes in with being a highly sensitive, scalable, and fast approach to reproducibly identify off-targets in the genome. So for the next few slides, I'm just going to quickly run through an introduction to the technology and some of its applications in therapeutic development. So our INDUCE-seq solution is ultimately a scalable platform for mapping on and off-target break events throughout the genome. And actually, this is a very quick process, and we can get from sample to answer in really just a couple of days. Importantly, INDUCE-seq is a cell-based technology, allowing us to identify off-targets directly in a clinically or a therapeutically relevant cell sample that we're working in. And this is super important in a context of, say, a potential gene or cell therapy. We have an unbiased genome-wide measurement of all these potential sites, which actually gives us, while going very broad throughout the genome, we have a single nucleotide resolution readout of the events occurring where we're making the edits. The innovation around how INDUCE-seq works is in its novel PCR-free workflow that actually helps to eliminate bias introduced by the kind of processing induced biases from PCR. And what's really different about INDUCE-seq is it's actually applicable very broadly to different kind of modalities in this space, pretty much to any gene editing system, but also any kind of therapeutically relevant cell type, whether that be cell lines, iPSCs, and actually a range of primary cell types as well. So INDUCE-seq's best-in-class sensitivity and scalability is really enabled by our novel flow cell enrichment methodology for capturing and labeling these breakends. So the process works by starting with a gene editing experiment where we're inducing our DNA breaks via, say, a CRISPR system in cells. We then capture these double-strand breaks in situ from a sample of cells, directly labeling the broken ends inside the cell nuclei in a sample of those cross-linked gene editing cells in a pretty standard 96-well plate format. We then move from a cell-based part of a workflow into a DNA-based workflow where we then go and label all of the other exposed ends with a second sequencing adapter, and we then read these out and enrich them directly onto a next-generation sequencing flow cell to capture and then sequence those positions of DNA. And what this does is it gives us this quite unique readout now where every single read generated from the sequencer actually corresponds to a single labeled break from that starting cell material. And what this allows us to do is generate an unbiased map that reveals the positions of the on and off targets throughout the genome, which ultimately, when it boils down to it, is a list of all of the potential sites that we observe high levels of break induction and where they are located in the genome. And it's really, this is actually quite a difficult challenge, is getting to this quite simplistic view of where these sites are and understanding, the potential risks of those. It's really, this is the actionable insight needed to develop safe genetic medicines, is having this understanding. So INDUCE-seq is actually, as I mentioned, a single nucleotide resolution readout where we can now actually reveal the mechanism of the break events directly. While going very broad genome-wide, we can go and zoom in right at the break site and actually get to the exact positioning of the nuclease cleavage sites. And so the data on the side here is just some examples looking at a range of editing systems from your standard Cas9 nuclease on the top left, through to a range of kind of modified and different CRISPR systems like type 5 nucleases and even things like TALENs. We can also now apply this, though, beyond nucleases, and actually INDUCE-seq enables the evaluation of bystander effects and off-targets actually induced by next-generation base editing systems. And the data on the side that I'm showing here actually shows two different panels of data. On the left-hand side, this is our standard double-strand break labeling methodology for INDUCE-seq, where we're actually now detecting across a range of base editing experiments unintentional double-strand breaks actually induced by the base editor, and we can map the breaks actually to the position of the nicked strand and the target cytosine that's being deaminated. On the right-hand side, we now have a slightly different panel of data where we now actually have in development a modified version of our INDUCE-seq technology where we can now actually target and cleave out those deaminated positions. So in this is a CBE system on the right here. We now have a much greater enhanced signal where we can now see we're mapping high signal essentially with that target cytosine against the next strand. So I talked earlier about INDUCE-seq being relevant across a range of therapeutically relevant cell types, and this slide really just shows a range of the kinds of samples we've actually used both internally in our own R&D group, but also in working with partners. And actually, this is something of real importance for developing cell and gene therapies, because many of the kind of key off-target methodologies that people use today are either cell-free, meaning you lack that critical kind of cellular context in taking those measurements, or actually there are high levels of cell toxicity in particular cell types because of the integration process necessary to capture those breaks. So this is actually an area we're seeing real interest and uptake is around particularly things like non-dividing cells, where we can work directly in things like primary neuronal cells and iPSCs. And again, there's very little modification needed to our workflow. We can feed in pretty much any starting cell material. And as a company at Broken String, our goal is really to address needs of customers throughout the entire development pipeline. And so today we have kind of a couple of different commercial offerings. We have a service where we're really focused on supporting customers in the late stage lead selection process into the preclinical and IND phase, where we're narrowing down those final candidates for taking it through into the IND and actually generating that kind of deep characterization of the potential off targets that go into the filing directly. But we also now have a product on the market to be run in customers' own labs. This applies to these same stages, but where we're seeing now is real interest is actually bringing the technology upstream even earlier into discovery, because we can now potentially de-risk many of these assets actually at a very much earlier stage because of the ease of the workflow and the fact it can actually be leveraged at scale in a kind of automated and 96 format as well. The last line on this slide I just want to maybe talk briefly to is actually, we have aspirations to go beyond this, beyond simply measuring these sites and revealing where they are, but actually to then build into, move into the kind of design phase where we actually want to bring a data-driven approach to the design of these guides and use our extremely clean INDUCE-seq data to actually better predict how these kinds of guide RNAs we're designing actually might induce sites and essentially, build safety into the design of them up front, which is something that typically is only really addressed much later in the process. So for the last few slides, I'm going to just talk about a few different collaborations we're currently running in case studies where we're now applying this in different settings. So the first is a group called HESI CT Tracks, where we're actually now working as part of an international collaboration on looking at off-target editing. And ultimately, the goal of this group at HESI is really the evaluation and standardization, really, of technologies for the reliable, robust measurement of on- and off-target edits induced by CRISPR-Cas9. HESI is an international group, kind of a private-public partnership where we have a number of different big pharma members. You also have kind of regulatory groups like the FDA, the EMEA, and NIST in there as representatives, as well as a number of different academic institutions. And through this study, we worked across this group to essentially design a study where we had gene editing performed at 2 different sites, one at the AstraZeneca Cambridge site and one at the Novartis site in Basel, where we had completely identical experiments performed at 2 different locations, which was then brought together. We measured a range of different guides across this, which are common reference guides, including the BCL11A guide that went into the Casgevy product as well. And the goal here is to really benchmark the activity of these different guides with the technology and compare things like reproducibility and sensitivity across these experiments. So through this process, we actually established and have been ongoing and developing an approach for identifying and essentially evaluating these off-target sites. And the way we do this is by creating an off-target superset, where we actually are revealing all of the potential sites that might have been induced by that editing system. And again, this approach is really to be as comprehensive as possible with the kind of mantra of trying to avoid any false negatives. So we're aware there may be false positives in here, but we're trying not to miss anything. And again, all through the lens of getting to a comprehensive list that can be taken through for monitoring. In short, this essentially comprises looking for sites that are very close to real detected breaks that overlap sites that are very similar to the guide sequence, but also looking at sites that are just very high level. They're broken at much higher levels than the typical background that we see, because we're measuring all breaks in these samples, and we essentially combine these 2 sets into really a superset list where you have then these 2 different groups, where you have the 2 different categories and then those that are shared between the 2. And so this is just a snapshot of what was a huge amount of data generated from this study, where we took many of these different guides, and we actually followed up all the way through to mutational outcomes at every single site we monitored or detected here, with both IDT's RAMP-seq technology, but also a more sensitive approach called duplex sequencing. And the reason for this was because we really wanted to understand the relationship between the breaks that we are detecting now and what then those mutagenic consequences of those breaks are. And so, in short, this is what we found is these things correlated actually very, very closely indeed. It wasn't a linear relationship, but actually a power law relationship where ultimately where we see high levels of breaks, we always see high levels of mutation, but this drops off in a very rapid manner, and so it's not a simple one-to-one linear relationship. This is currently being written up and is going to be published, hopefully by the end of the year, at least on the bioRxiv, showing all of that, what was essentially a multi-year study where this is all mapped out. So in the final minute or so, I'll just talk about a collaboration we're actually currently running with the Innovative Genomics Institute, where we've now working with Dr. Fyodor Ernov's group and have set up INDUCE-seq at the labs at the IGI. And so the goal of this work is to bring INDUCE-seq into their labs, but also set it up as a way we can now address the detection of off-targets in a very rapid and scalable way. And in particular, if any of you are familiar with Dr. Fyodor and his work, there's this rare disease focus where we have to get to these off-target answers very, very rapidly, because ultimately there's like an N=1 kind of patient situation where this needs to be done very quickly. In this study, we've actually been working on a number of donor samples, human pluripotent stem cells, edited with a couple of different reference guides. And this is actually just a live collaboration where we're now working through the nomination process with their team, and we're going to be following up at all of these sites. So in the last slide or so, just to talk about where we are with our kind of offerings. So our platform is now available to run out of essentially any lab. So this is a complete sample to answer offering, where we have it entirely productized through the wet lab component, but also all of the software and analytics. Our solution comprises our INDUCE-seq library preparation kit, which essentially enables the labeling and sequencing of breaks in the genome. This is then followed up by sequencing, which would be run either in your own lab or at a service provider. We then also provide a software analytics platform, which allows the raw data to be processed and the identification of these lists. And then finally, we bring in a last step where we have an AI approach to kind of rank and score and filter down those sites into a set that you would be then nominating for monitoring. And this is, as I mentioned at the start, a pretty rapid process. You know, we can now, from editing, we can get to answers in really just a couple of days, meaning it's now feasible to do these kinds of editing experiments much, much more quickly. So, a final slide just on some acknowledgements from the people who have contributed to the work, and thank you everyone for listening.